we were discussing about how this dual nature of Rubisco is creating, in a way, some issues to our photosynthesis. And we see it as a problem, right? And I also recommend you to read the article on your eHub. So regarding this, whether it's actually a negative thing, like 100% negative thing, or it's just it has some negative minor or like a medium level negative effects. And if you think about plants' perspective, so they're not going to stay quiet and let something interfere, inhibit what is doing. Because here as human or as scientists, we are trying to find solutions. But plants also, they're perfect, well, I can't say perfect, but they're darn good scientists themselves. So they identified this problem, and so they were thinking, okay, how we can deal with this love and hate relationship between oxygen and carbon dioxide with the Rubisco enzyme. During the evolution, what happened was some plants, they came up with, some people call it as syndrome, different mechanisms in order to overcome this issue. Okay, so the most the most interesting and cool thing is, when plants evolve, so they evolve in different places, and they have a different isolations and different environmental conditions surrounding them. So what we observe once we because now we study so many different species about their physiological characters, anatomical characters, as well as the genetic characters. What we realize is at least in 14 different places there are new mechanisms were evolved from conventional C3. Okay, and I think probably during your A levels, I think you learn about C3, C4, and CAM. But nature, it's not simple like that. So the cool thing is, some plants, of course, they became C4 plants, very clear, C4, and then some stayed as C3. Some plants, what happens is, so they start their life as C3 plants, and with time, they will become CAM plants. And then some plants, so they start their life as C3, and then they get some C4 characteristics. And this is so awesome, because some so they look like, say, normal C3 plant. Imagine they're changing the whole photosynthesis machinery into CAM during the latter part of their life. Okay, so don't you think it's cool? I mean, I think it's super and super cool. Okay, and so this, if you look at this, the circular phylogram, you can see all these different colors represents different carbon dioxide concentrating mechanisms found in these plants. Okay, the ones without circles, those ones are the C3 plant. You can see different places, different lineages, different mechanisms, and they are independent from the other reactions. So that means the evolution happens in different places, but the outcome, sometimes the outcome was similar, but they are they never seen these plants each other during their evolution. Okay, so in very simplified diagram, I can show you how the CAM evolution happened and how the C4 evolution happened. So it all started with C3 ancestors, and what, as you can see here, okay, And the camp plants, I think by nature, they are very succulent. Okay, if you take aloe vera and other cactus like plants, they are very succulent because they are trying to preserve their water. Okay, so therefore we think during the evolution, the camp evolution, the first the appearance of succulents, the appearance of succulents started at the beginning. Okay, because in order to increase the water capacitance. Okay, and then what is going to happen? What happened was basically the succulent level increased, and also as a result, these mesophils, they are going to get very tightly packed. Okay, that is one of the natures. I will show you section of camp plant probably towards the last part of this lecture. And you can see it also during this process, they recapture some respiratory carbon dioxide by PEP carboxylase, and then they develop camp cycling, and also they came up with this circadian control. I think you are familiar with circadian. Circadian means basically day and night. Okay, so once all these things came into one picture, so then they got the 100% camp capability. Okay, and if you think about the C4 evolution, let me pick a different color. Let's say purple. Yeah, and the C3 ancestors. So what happened was, so some of the plants, they reduce the mesophil to bundle sheath ratio. Okay, and this during this process, as you can see, the expansion of bundles sheath cells started, and also there was a reduction in mesophil, and eventually it was basically converted to Kranz anatomy. That is something I'm going to show you next. And then if you think about the biochemistry side, you can see the glycine decarboxylase basically restricted to the bundle sheath cells, and also the rubisco was enhanced in bundle sheath cell, and there were reduced mesophil cells. Okay, and then PEP carboxylase enzyme level was increased in mesophil cells, and then towards the latter part of the evolution, the PEP carboxylase was found only in mesophil cells, and rubisco was only in bundle sheath cells. And once it acquired all these characters, they became 100% C4 plants. Okay, so see how cool it is. So here the anatomy played a major role. So that's why we call as the C4 photosynthesis as an anatomy first approach, and the chem, so they were more towards the biochemistry side. That means basically biochemistry first. First, let's look at the C4 photosynthesis, especially the anatomical modification. As you can see here, the C4 plants, so they have a different leaf architecture. They have mesophil cells, and the most interestingly, they have very nice set of bundle sheath cells. Okay, so this is what we call as Kranz anatomy. Okay, so it was discovered by a scientist in Germany. His name was Kranz, so that's why we call it as Kranz anatomy. And let's see what is happening between this, happening in mesophil cells, and between mesophil and bundle sheath cells, and then the bundle sheath cells itself. Okay, so look at this diagram. Okay, so you have the external atmosphere, and this is the external region, and you can see here, the carbon dioxide molecule was captured by the mesophil cells, and then it is going to dissolve and will come by carboxylic acid, right? I'm not good in chemistry. And then this one is going to basically react with PEP carboxylase, and then what is going to happen, it will be converted into malate. Okay, and this malate is basically going to the bundle sheath cells, and through a diffusion barrier, and then it is going to go into the mitochondria, and with the help of NADP malic enzyme, it will be converted into pyruvate, and this pyruvate is basically going back to the mesophyll cells, and then inside the chloroplast it is going to basically react with pyruvate phosphate dikinase, and then it will be converted into phosphoenol pyruvate. It's a C3 compound, and then it will basically react with HCO3 minus and then create malate. Okay, so this is one side, and the most interesting part is you can see now it is dropping this carbon dioxide from outside right onto the chloroplast located in the bundle sheath cell. So now imagine, so there are many mitochondria, right? So they're like putting carbon dioxide all around this chloroplast. So that means the CO2 concentration is going to get really high. So that means when the Rubisco, when it acts, what is going to happen? So it has more carbon dioxide than oxygen. So therefore, it's very high level of carbon dioxide concentration around this Rubisco, and that means so you can basically forget about the port respiration rate is going to get reduced into a very, very low level. Okay, and if you think about the plants, remember I told you these C4 mechanisms and also CAM mechanism and C3 CAM mechanism, they started in different lineages. So therefore, as a result, we see a diversity when it comes to the different C4 acid decarboxylation in chloroplast of our bundle sheath cells. Let's see what are those different decarboxylating enzymes involving these processes. If you look at this table, you can see, so there are at least 3 major decarboxylation enzymes. One is, as you already saw, NADP malic enzyme. That's the first one. And then there's NAD malic enzyme, and then PEP carboxykinase. Okay. And what is going to happen is once they react, so they are going to basically transport the C4 acid from mesophyll cell into bundle sheath cell for the decarboxylation. And this first stable compound involved in this process for the NADP malic enzyme is malate, and then the other 2 enzymes, it is going to be aspartate. Okay, and then if you think about the C3 acid move from bundle sheath into the mesophyll for the carboxylation, for the first one it is going to be pyruvate, and then if you think about the NAD malic enzyme, it's going to be alanine, and for PEP carboxykinase, it is going to be either alanine or pyruvate phosphoenol pyruvate. Okay, so therefore there are differences when it comes to the what acid that was transported into bundle sheath cells, and then what C3 acid moved into mesophyll cells by bundle sheath cells. So there's a difference. And if you look at the sorghum and siamese, they have NADP malic enzyme. And if you think about the Cleome or triplex plant, they have the NAD malic enzyme. And then the grasses like panicle, so they have PEP carboxykinase. So therefore remember, although maybe most probably you learn about only one enzyme, but there are more than one enzyme involved in this C4 process. And when we talk about our Kranz anatomy, I already explained the most significant thing about Kranz anatomy is because of this anatomical adaptation, it is going to increase the CO2 concentration in the bundle sheath cells. And this is 10 times higher carbon dioxide concentration compared to the carbon dioxide level outside with the atmosphere. And as a result, it facilitates the efficient accumulation of carbon dioxide in and around the vicinity of chloroplast rubisco. And as a result, remember, the dual nature is going to get interrupted. So therefore, the photorespiration rate is going to get reduced to 2 to 3% of the total photosynthesis. Okay, so therefore, see, plant experience a problem, and then plants came up with the solution with the beauty of the evolution. Okay, so if you compare the differences between C3 and C4, so there are a bunch of differences. One is PEP carboxykinase is located in the mesophyll cells. Rubisco is located in bundle sheath cells. And if you think about the decarboxylases, they are found in different intercellular compartments of our bundle sheath cells. Okay, and if you think about the NAD or NADP malic enzyme, NADP malic enzyme, it's located in chloroplast, and NAD malic enzyme is located in mitochondria, and the PEP carboxykinase is going to included in the cytosol. Okay, maybe I mispronounced. So this is PEP carboxykinase and this is PEP carboxylase. Okay, and moving forward, if you think about the anatomy, our mesophyll cells, they contain randomly arranged chloroplast with lot of stacked thylakoid membranes. And in contrast, if you think about the bundle sheath cells, they contain more concentrically arranged chloroplast, okay, and they exhibit unstacked thylakoid, okay, because now they are facilitating processes in like two different places. So therefore, they basically change the structural content of chloroplast. And so this is like a very simple schematic diagram of like the C3 and C4 anatomy plants. But now like everyone is like I think okay C4 is Kranz and that is it. But in reality, remember I told you C4 originated in different ways, and you will be surprised that some single cells, they can do C4 photosynthesis. Okay, they don't have mesophyll and bundle sheath. Instead, so they basically separate the every reaction inside a single cell. See how amazing it is. Okay, so let's if you look at this diagram, so here we are talking about single cell C4 cycle. The atmospheric carbon dioxide is going to basically enter into the external side. And then what is going to happen is basically, so there is like spatial isolation. So that means inside the single cell, there are like inside external side and internal side. And what is going to happen is like earlier, the C3 compound. The C4, the C4, what do you call them? The first stable intermediate product is going to be produced, and with the help of the enzymes we discussed, and then this C4 will be compound will be moved towards the internal side of the cell, and then it is going to release the carbon dioxide. And then what is going to happen is basically, so here the carbon dioxide concentration is going to be higher than the external atmosphere, and then when it releases carbon dioxide, it creates a C3 molecule and it is going to the external side. Earlier we saw two different cell types, so here now it's happening inside the single cell. And one example is diatoms, so they can do a single cell C4 cycle. Okay, so whatever the carbon assimilation happen in the internal side, they will be basically transferred into the vascular tissues in the multicellular plants. But in single cell plants, the unicellular organism, it is happening. I mean, basically that carbon assimilation also happening inside the single cell. So therefore, if it's a land plant, so they basically make our carbon assimilation and then transport it to the other places. If it's unicellular, one cell organism, it is going to, the metabolism is going to happen inside the same cell. And so there's some very, very cool electron microscopic images of these mechanisms. So one, as you can see here, if you look at the Sweda plants, if you're doing marine sciences, I think this is one of very cool plants people study. And as you can see here, they have like elongated cells. And then as I showed you earlier, there's a cytosol diffusion barrier, and internal region you have more concentration of carbon dioxide compared to the outside. Okay, so therefore they are spatially isolated. And in Venetia, as you can see here, so it's like more circular type. And what is going to happen is, so they have the chloroplast arranged in a different way. So that way, basically, some chloroplast is going to do the job in towards the external side, and then it will be transferred to a internal side where they have a different chloroplast architecture. Okay, so see how plant came up with two cell system as well as one cell system to achieve the same goal. Okay, so that's all I'm going to talk about C4 photosynthesis. And so there will be some reading I will tell you at the end of the lecture. And let's jump into the CAM mechanism of acylation acid metabolism. And this one originated more than 35 million years ago. And the objective was to, as I mentioned earlier, conserve water in habitats where there is an insufficient amount of rainfall, and therefore it is not enough, or again it's insufficient for crop growth. Okay, so the leaves of the CAM plant, they are evolved in a way in order to minimize the water loss. So therefore they have very thick cuticles and huge vacuoles, and then their stomata is basically hidden, and they have very tiny apertures. And remember, because of the succulent layer, the mesophyll cells are very tightly packed. So therefore, it enhances the CAM performance by restricting the carbon dioxide loss into the environment. Okay. And in all CAM plants, the initial capture of carbon dioxide into a 4-carbon compound happened during the night. Okay. And then the posterior incorporation of carbon dioxide into carbon skeletons happen during the day. Why? Because daytime is really hot and very dry. So therefore it loses water. So because if it starts to opening its stomata, and then carbon dioxide will be lost into the atmosphere. So what they do is, at night they basically take carbon dioxide and then convert it into 4-carbon molecules. And then during the daytime, they keep the stomata closed to prevent the water loss. And then they do the next part. If you look at a schematic diagram, as you can see here, during the dark, they have the stomata open. And this open stomata are going to basically permit the uptake of carbon dioxide into the plant. And then it will basically dissolve into carboxylic acid. And so what is going to happen is PEP carboxylase is going to convert into oxaloacetate, and so then oxaloacetate will be converted into malate with the help of NAD malate dehydrogenase enzyme, and this process needs NADH as an input, and then it is stored as malic acid inside the vacuole. In order to make our oxaloacetate, the phosphoryl pyruvate is coming from the metabolism of starch into triphosphate to phosphoryl pyruvate inside the chloroplast, and then eventually at night now you have a storage or a reserve of malic acid inside the vacuole. And when light is there, what is going to happen is the stomata, they are going to close because otherwise they will lose the water. So now the malic acid stored inside this vacuole, as you can see here, will be basically coming into the cytosol, and then this malate will enter into the chloroplast, and then inside the chloroplast this malate will be converted to pyruvate with the help of NAD malic enzyme, and this pyruvate will basically go through the process of making starch, and then this the whatever the carbon dioxide that is coming out from this malate, so that will be used for the Calvin, Benson and Basham cycle, and then it will be also converted to starch. Okay, so this is what's happened in the crustacean acid metabolism. It is basically, during night time, it captured carbon dioxide and then converted to C4, and then it will be used during the daytime for the production of starch, and there's minimal carbon dioxide captured during the day. Okay, so now you see there's C4, there's CAM, and then also we talk about single cell C4 photosynthesis. So now next question is what to do with outputs of our photosynthesis. And as you can see here, whatever the products coming out from our the photosynthesis, they will be basically one part will be stored as a starch inside the chloroplast, and then some triphosphate, they will come into the cytoplasm, and then they will be converted into sucrose. And remember, sucrose can move, but starch can't move. So therefore, the sucrose is the mobile, the carbohydrate that will be transported across the plant, as you can see here. And during the night time, so whatever the starch that was stored in the chloroplast will be basically move into the cytosol after converting to maltose and glucose through the maltose glucose translocated, and then these hexophosphates will basically converted into sucrose, and then that sucrose will go into vascular tissues, and some will be used for the growth and development of the plant, and some will be stored for for us as well as the survival of the plant. Okay, so therefore that is the fate of the photosynthase coming out as a result of photosynthesis process. Okay, so that is all I'm going to talk about the photosynthesis, and specifically here we talked about the carbon concentration or the carbon reactions, and especially the carbon concentration mechanism happening inside different plants. Now the question is, okay, so now we know that C4 is more advantageous, because CAM is CAM plants are not very efficient when it comes to the photosynthesis, because they are adapted to one specific environment. But if in contrast the C4, so they are very productive, and therefore they can make a lot of yield, and then it is very beneficial for us because the efficiency is high. So now what is your so probably during today's lunch you're going to eat rice, right? For us, that rice is the main staple food, and unfortunately, fortunately, I don't know, it is a C3 plant. So therefore now imagine, because now our population is going to reach 10 billion in 2040. It's already 8 billion, and remember there is big organic fiasco, so we didn't even have enough rice to eat, so because of lack of fertilizer. And so that is one thing. And also we are experiencing droughts, remember, earlier this year. So there's a huge drought, so therefore that affects the ultimate production. But imagine if we can convert rice into a C4 plant, that will be amazing, right? So there's one big project going on, International Rice Research Institute. It's called as the Rice C4 project, and they received 4.2 million 4.2 billion research grant from the Bill and Melinda Gates Foundation, and they are given the task to come up with the C4 rice plant. So now you need to think very carefully. Think about the rice, the architecture, the appearance of rice, the mesophyll cells, and they are not very well separated into bundle sheath, and then the mesophyll cells like in corn or the maize plant. So therefore, like, now you need to come up with a strategy how we are going to incorporate C4 mechanism into this in this C3 system and make it successful. That is something like, we the scientists, they were like, puzzled, okay, because I mean, it is difficult, but like there are different approaches. Because there are minimal requirement to do that kind of a change. Okay, so let's see what they try to achieve through the RISE C4 project. Okay, so if you look at this diagram, I think I already uploaded that paper into eHub as well. If you're interested, please read it. Otherwise, just like, focus on what I'm talking about. So what they did is like, okay, so in order to make that kind of a system, because rice already have bundle sheath cells, they are not specifically involved in this the same thing as the C4 plants. Instead, they are like, as far as I remember, they are involved in more like sulfate and like, nitrogen metabolism. So that is kind of their focus, but not to do C4 photosynthesis. Okay, so their approach was to basically introduce a set of enzymes into this mesophyll cells specifically and bundle sheath cells. So that way, basically, you can incorporate some aspects of C4 photosynthesis into a rice plant. And also, only putting enzyme is not going to work, because remember, so there's always exchange between mesophyll and bundle sheath cells in the C4 plant. So therefore, they also focusing on different transporters. So that way, so basically, you can facilitate the transfer of different molecules into the correct place in order to this process to happen. And this project is probably like 6 or 7 years old right now. And so they got some success, because it is not simple as it seems. And probably you're thinking how they are trying to do it. They are trying to do it with the help of biotechnology. And one group, so what they did is basically they made like, individual plants carrying this like, natively introduced enzyme. Okay, so therefore, like, we are introducing 5 enzymes, and then they have like, 5 individual one set of cultivar, so like, hybrid not hybrid, sorry, cultivar, like, pure line containing this one specific gene, and then conventional method. So they were basically crossing these plants and then purify it, like, so that takes like, remember, you already did the breeding course, right? So therefore, like, it takes like a 7-8 generation. So therefore, like, it takes like probably 7 years to achieve that. But somehow they incorporate like, most of those things, but it's a very time consuming approach. So one other group, so they were thinking like an even more cool way. So they decide, okay, we are going to basically put all these genes, remember you learn about vectors, into like a one vector, and then we will basically introduce into one line. Okay, so they were successful in that as well. So instead of like, going through like 7 years of like, nightmare, so they can do it within like, one run. But like, remember, introducing is easy, but we are expecting this one to express in this specific place in this amount. So therefore, like, it is not yet like completely achieved. Okay, and then the most successful approach came very recently. What they did is like, this is my favorite area as well, because I study like, gene promoters. Because to tell you, like, gene promoters, there are like, small patches of sequences, what we call as elements, or we call it cis regulatory element. So this cis element, so they are pretty cool. So let's say if you want to express, let's say you have a favorite gene. Okay, let's say you like to eat purple rice, and then you want to make a plant, like, where its seed is going to make purple color rice seed. So now how you are going to do it? So let's say you have the purple gene, and okay, of course you are going to put it into like, the the rice genome, and then what is going to happen? The whole plant is going to be like purple. For like maybe chloroplast will cover it, but like that purple is going to express. The anthocyanin is going to get expressed inside the whole plant. So therefore, like then it's not going to give you the purpose, because you want to eat purple color seeds, not like you're not going to eat like strona, unless you're like goat or something. So therefore, the beauty of this promoter, like I told you, like there are tiny elements. So these things basically they are going to determine when, where, and how much that gene is going to express. Okay, so let's say if you want to express your purple gene only in the rice seed, so you put a promoter element, or you introduce a promoter element, because some promoter element they only let whatever the corresponding gene to express near the inside the seed. Okay, so that is how cool it is. So likewise, what they did is like basically they identified a set of promoters. So these promoter elements, they are basically infused in front of those like different photosynthesis related gene. Now what is going to happen is, and they put it into like different what we call as construct. So therefore now we like basically do the transformation. Although they are in like every cell, they will be only expressed in like if in that bundle sheet cells, and the other set is expressed in mesophyll cells. Okay, so see how cool it is. So that means, so now we can decide, okay, how much and what cell type these photosynthetic enzymes are going to work. So this is like one of the very latest papers. It came in out in 2022, and they were like very successful doing that. So therefore, I think probably within next 5 or 6 years, we will be able to get like C4, or like at least near C4 rice lines for us. Okay, so fingers crossed. So therefore, like so there's a lot of hope left for us. Okay, so and then probably like when you guys become scientists, I hope to see like more C4, like more photosynthetic engineering done by you. So therefore, like I'm keeping my fingers crossed. Okay, so with that, I think I'm going to conclude my lecture. And next week, Professor Chandrika Perra is going to start the respiration section. As always, if you have any questions regarding today's lecture, please post your questions under lecture 3 feedback. As I always tell you, so please give your please post your questions if you have any concerns, and if you have any suggestions, or if you have any critiques. So please feel free to use the lecture feedback section to do that. And I will try to upload a glossary that contains different terms related to the photosynthesis, and within next couple of days. And there will be one forced reading, so where you're asked to read this paper. Please read this paper. So that paper is basically talking about what are the challenges we are facing, and what approaches we can take in order to improve our crop by changing their photosynthesis from C3 to C4. Okay, so please read that paper. Some parts you might not understand, but don't worry too much about it, but at least read it and then grab the most important points. So that's all I expect from you. And if you I know there are some people, they like to read more, because I told you that C3 and CAM evaluation is very interesting. I already added fun reading talking about C4 and CAM evaluation in our eHub as well. Okay, so with that, I'm going to conclude the lecture, and I'm hoping to see you, because I have only one lecture left, and that's towards the the very last week of your semester. So therefore, until then, I'm saying goodbye to you, and you guys have a very good and wonderful day. Thank you.